the eyes don't see, it's the brain which does that. The retina is quite delicate. For you to actually fix that tissue, embed it in wax, and get slides from which you can view, that was an uphill task. Let's have a balance on intake of folate, because we have established that an excess and also a deficiency can cause anatomical disruptions at a microscopic level in the rats.
Welcome to the EAJNS podcast. Today, we will be discussing a fascinating article which explores how maternal folate levels during pregnancy affect neural development of retinal tissue in rat offspring. This study highlights something very important in both medicine and public health: the deficiency and excess of a nutrient can have consequences. Today, we're joined with Wallace Munyiri, one of the authors of this article. Thank you and welcome.
Thank you for having me. It's a pleasure.
As we begin, we'd love to understand the journey behind this study. What drew you to study the retina as a component to understand the neurodevelopment?
To start us off, allow me to first tell the public what the retina is, for those who might just have an idea, or those maybe who don't have an idea of what the retina is. So, the way our eyes, in quotes, see is because they are able to convert light energy to electrical energy, which is now the nerve impulses. The part of the eye which is responsible for doing this is the retina, which is found at the back of our eye. The reason I said in quotes see is because it's the eyes don't see, it's the brain which does that. So the retina, it is a extension from the brain. They all come from the same primordium, from the same, in quotes, parent, the neurotube. It is known that folate deficiency causes neurotube defects, but not much has been done to investigate how folate deficiency or folate excess affects the development of the neuroretina. So the retina is further divided into two parts. We have a neuro part and what is known as the retinal pigment epithelium. Let us just say the neuro part. So my research focused on the neuro part, and that is what we were investigating. What sparked this interest? Maybe when I was a first year, I was always interested in embryology, learning how we were formed in our mother's wombs, and that, I think, sparked the interest to understand how our mothers, when they eat, how does that dietary habit affect even the outcome. And that is what sparked the interest to now study the retina.
So folate is widely discussed in relation to neural tube defects, but much less so in retinal development. What is the proposed relationship between neural tube defects due to folate deficiency and retina development?
So as I mentioned earlier, they all have a common parent. The neurotube gives rise to the spinal cord, the brain, but also the neuroretina and the optic nerve, the nerve which enables you to, in quotes, see. If it has been established that folate deficiency causes neurotube defects, then wouldn't it be more interesting to now investigate how folate deficiency affect the development of the retina? Also, excess folate has been shown in studies that it causes um, neurodevelopmental disorders in the cerebellum and also in the cerebrum, and it has been associated with the autism spectrum disorder, the excess. So it shows you that both deficiency and excess have an effect on the development of the neural tissue, but current studies had not investigated the morphological differences that would come about due to varying levels of folate in the neuroretina, and this is what this study aimed to investigate. Allow me to just say that.
Yeah.
Yep.
I'm very interested to understand how you came up with this research question. Was there any academic or clinical incident that sparked your interest?
Okay, so maybe it was more of academic. I'm not an ophthalmologist, so I won't say it's from clinical experience. Allow me to just say that my mentor, Dr. Mwachaka, he investigated how these levels, varying levels of folate affect the development of the cerebellum. Now, I had the idea that since this cerebellum, neuroretina, all of them have a common parent, a common primordium, then it would be interesting to also investigate how the same varying levels of folate during the perigestational period, the period around pregnancy, would affect the outcome or the development of the neuroretina. So it was more of an academic interest, not a clinical interest.
So this whole research project has been a journey for you. Take us through this journey and also help us explain your study design in a very simplified manner for our general audience.
All right, so I want you to imagine you have 4 groups of rats. So we took 4 groups of rats, female rats, 2 in each group, so maximum of a total of 8. Then they were exposed to varying levels of folate for a period of 2 weeks. So the diet groups were as follows. We had a folate deficient diet group where they did not receive any folate in their diet, a control group, a supplemented group, and a folate excess group. So those were the 4 groups. So these rats were fed on these diets for a period of 2 weeks, after which mating was done, and then the diets were maintained throughout pregnancy and throughout lactation. Then the offsprings were sacrificed at day 35, after which we obtained their eyes, and from the eyes we obtained the retina and studied the retina under the microscope. I hope that is a simplified manner.
Thank you. That makes it very easy to understand.
Yeah.
Now, your analysis focused on the central retina. Why is this region near the optic disc particularly informative when assessing developmental changes?
Now, the reason I chose to study the area of the central retina just next to the optic nerve is more of a methodological issue, so that you can maintain uniformity across the groups. You want to, when investigating the 4 groups, you want to have an area of reference. You don't want to just study any area because the retina is big. So you want to see, does this varying levels of folate affect the same area of the retina across the 4 groups? So we maintained the area of 100 micrometers from the optic disc, and that is the area which we studied. So it's more of a methodological issue to maintain uniformity when studying the differences across the 4 groups.
Now let's talk about the heart of your paper, which are the results. One of the most striking observations was that both folate deficiency and folate excess disrupted the retinal morphology. Did these results surprise you?
The truth is they did not.
Okay.
because previous studies had shown that deficiency and excess adversely affect neurodevelopment both in the cortex and in the cerebellum. So I was not much surprised to find that supplement the excess folate group had also a disrupted retina. Maybe what surprised me is they were almost identical, because in the other neuro tissues, you'd find that excess and folate, as much as you're talking about disruption, they are of different types, but here in the retina, they are almost identical. So I think that is what surprised me.
Take us through how you conducted the study, came to the results, how you observed these results, what methods you used.
Okay, in a simplified manner. So remember, we go back to the methodology section. Once we got the eyes, we prepared them for histology, where we stained using a stain. So you use a stain to see tissues. You can't see tissues under a microscope without staining them. So we stained them and then observed them under a microscope under different power lenses. So from at a low magnification and maybe at a higher magnification for finer details. So that is how we saw the differences. We also took photos of the differences and analyzed them using various analyzers, if I can put it that way.
What was the most surprising part of your results?
The most surprising, maybe to say the control group and the supplemented group were also identical. So it seems that the supplemented group and the control group, which is the control group, we give them folate, which is recommended on a normal daily intake, and the supplemented group, they almost had identical findings. And I think this cements the idea that we should not take folate for granted. It should be there in our diets every day, and even for maybe mothers who are planning to get pregnant, it should be there as a daily requirement. So the control group and the supplemented group being identical, I think that was a quite interesting finding.
Your discussion proposes several biological mechanisms underlying these changes. You describe homocysteine accumulation as a possible shared pathway for both folate excess and deficiency. Why is homocysteine so damaging to retinal tissue?
Okay, so another technical term, homocysteine. So take homocysteine as a bad molecule. It's a bad molecule. It should not accumulate in your body at all. And homocysteine is found in what is known as the methionine cycle. Another technical term. Maybe I should decrease those. So for you to convert homocysteine to methionine is a good guy. So imagine you're converting a bad molecule to a good molecule. For that conversion to happen, you require folate. So if folate is not there, homocysteine, the bad molecule, now accumulates in the body. Why am I calling it bad? Because it damages your vessels and also produces what are known as reactive oxygen species. Now take reactive oxygen species as bullets which penetrate the cells and they kill the cells. So if homocysteine is in excess, one, what it does to the retinal vessels, those vessels giving oxygen and nutrients to the retinal cells, it damages those guys. So what will happen? The retinal cells will die. Or if that does not happen, homocysteine itself produces bullets the reactive oxygen species, which can now directly kill the retinal cells. And the nervous tissue is very sensitive to the reactive oxygen species. So that is how homocysteine is important in retinal development.
Thank you so much for your analogy. It makes understanding the whole process a lot more simpler. If you were given access to advanced techniques, which ones would you use to better your study?
Okay, so thank you for asking that, because we just used the basic histological stains. But if I had access to maybe more advanced techniques, I'd go for one, immunohistochemical stains, because these ones can enable me to know what exact cells are more affected, or do we have a certain population of cells, because the retina is made up of various cell types. We have neuronal cells, the photoreceptor cells, those cells which now convert light to nerve impulse. Then we have the neuronal cells and the association cells. So immunohistochemical stains can really help you to know of these cell populations, which one is affected more, or which one maybe are even more resistant to changes in folate. Secondly, I'd study how the retina is affected by varying levels of folate across different timelines. So I just used the postnatal day 35, but it would be interesting to know even how is it affected even before the child is born, or the rats are born in this case, at birth, and maybe now postnatally, which is already established. So before the prenatal, how is the prenatal? How does it look prenatally? So I think that would be a good way to investigate across different timings and using immunohistochemical stains.
What was the most challenging part of your study?
Challenging, I think it was the histology bit. The retina is quite delicate. For you to actually fix that tissue, embed it in wax, and get slides from which you can view, that was an uphill task. The other tasks regarding rats will always survive, but the technical bit was now to get these slides. That was an uphill task, but I am glad that I was able to climb that hill and manage that task.
That is amazing. Now, to a more positive note, what was your most memorable part of your study?
Most memorable of the study, maybe of the experience, because when I was doing the study, we used to, there's a colleague of mine known as Grodia. We used to go to the histology lab as a team and help each other, maybe to even come up with the slides. And yeah, that's, I think, and maybe create a bond, a friendship bond, which even lasts today. So I think that's the most memorable part, and also with the other colleagues like John and Sammy and many others. So I think the memories are more of people, not more of the study.
Yeah.
So it's the friendship which you form along the way during the study. Those are the ones which you tend to remember the most. I have never dreamt of myself doing that study, or maybe being with the rats. It's more of with the people whom you met along the way.
That's beautiful.
Thank you.
Now let's talk about what this means beyond the lab, what your study means beyond the lab. Folate supplementation is strongly encouraged during pregnancy. How should clinicians interpret your findings without discouraging appropriate supplementation?
So yes, this study does not in any way wish to discourage folate supplementation. The set guidelines of folate supplementation are put in place, and the current study does not dispute that at all. What the study does, it's a hypothesis generating study, which questions, it just raises the question, does excess folate have an effect on development of the neuroretina or neuronal tissue? And how can this even affect the functionality of the eye later on in life? So it's a hypothesis generating question. To the clinicians, they need to still advise on folate supplementation, but with a caution on overdosing, unless it is clinically indicated, as it is in mothers who have had who have maybe given birth to children with neurotube defects. For those ones, it's recommended that they take an excess of folate to prevent this from happening. But if it has not been clinically advised, maybe you might want to hold your horses and not give an excess.
Yeah, which is very important. And your study highlights what excess folate does.
Yes. On the histomorphological structure.
Yes.
Yeah.
Thinking ahead, do you think these histological changes could translate into functional visual deficits later in life?
Okay, allow me to just say that we investigated the anatomy. So as an anatomist, I'll say that varying levels of folate for sure caused anatomical disruptions and differences. Now, regarding the physiology, I am naive. I don't have knowledge. So maybe as a recommendation for future studies, maybe they can do electrophysiological studies to see how these varying levels of folate could affect. So maybe a follow-up study. This time, maybe they won't sacrifice the rats. They'll now do conduction studies, maybe conduction studies in the optic nerve to see do we have functional differences? Because what I've established is we have anatomical differences. So maybe my view on that is we need more studies to actually investigate it.
You've proposed several directions for future research. What excites you the most about the next steps? Studying different developmental stages or using cell-specific techniques?
Both excite me because, as I said, timings: prenatal, at birth, postnatal. Cell-specific immunohistochemical stains. So I think both ways, both are exciting. Allow me to just say that.
If the listeners were to take home one message from your study, which one would it be?
So my take-home message would be focus more on balance than excesses. So let's have a balance on intake of folate, because we have established that an excess and also a deficiency can cause anatomical disruptions at a microscopic level in the rats. I think balance over excess would be my one take-home message.
Thank you so much, Wallace, for your very insightful discussion today. To our listeners, thank you for joining us today. You can access the full article on our website, EAJNS.org. Be sure to read the article and continue the conversation online. Until next time, thank you.